How big could COYA get? I think we could be a multi-billion valuation company. We have a good path with the FDA to get approved. Based on all the things I've told you, today we should be a 300 to 500 million cap company.
Welcome to The Upside with Lou Basenese. When most investors think about investing in biotech, they think of cures for cancer because those diseases are particularly nasty and claim life very quickly. But there's an even bigger opportunity and need when we talk about neurodegenerative diseases. I'm talking about things like Alzheimer's, Lou Gehrig's disease, dementia. It's estimated that there's 50 million people worldwide suffering from one of these diseases right now. And I'm fortunate today to be with the CEO of a company that could be providing a paradigm shift in how we treat some of those diseases. So welcome to The Upside, Arun.
Thank you, Lou.
CEO of COYA Therapeutics. Pleasure to have you here. I always start with what's the big breakthrough that COYA is unlocking to maybe change the path of treatment for these diseases?
Thank you, Lou. Thanks for having me on the show. What COYA wants to do, what we are excited about doing, is to make a difference for patients with diseases like ALS, frontotemporal dementia, diseases that today are really no options for these patients. Average ALS patient dies within 3 to 5 years of diagnosis.
Wow.
And FTD has no approved therapies with rapidly declining quality of life and lifespan. What we're excited about is that we have a strong scientific foundation through which we feel pretty good that we can make these diseases livable diseases. What I mean by
livable, stop progression where it's at, maybe reverse progression. What do you think is possible?
Stop progression is our goal. So if we can keep patients from not declining any further, just imagine that world. If a patient, for example, is walking with a cane today, and at the best case, we can keep them continuing to be there.
Right.
I think they'll take that any day.
No, absolutely. I mean, I had a grandmother who had Alzheimer's for 17 years. And so physically she was in great shape, but then mentally it just was very diminished over time. And she was a shadow of herself very quickly. So you're talking about early detection and being able to arrest progression for multiple neurodegenerative diseases. How do you do that? Like, what's the breakthrough that COYA is doing, that an approach that no one else is doing?
Yeah, our breakthrough is in our science. So we believe that if you address neuroinflammation, if you want to imagine it like a fire, if you can put out this fire, the inflammation, it'll stop the progression of almost all these neurodegenerative diseases. Now, your question may be, how do we put out this fire?
Yeah.
And that is through regulating something called the regulatory T cells. And I'll get a little bit more into it.
Yeah, we don't want to dive too deep into science right out of the gate.
What I want people to know is that, first of all, the Nobel Prize in October went to one of our scientific advisory board members, Dr. Shimon Sakaguchi, for his discovery of these Tregs and its application in diseases. And then Dr. Stanley Appel, whose shoulders we stand on, did a lot of work showing that when these Tregs are dysfunctional, bad things happen and neurodegenerative diseases progress.
So it's all downstream of that fire. It's what happens, the damage that's caused by this inflammation or fire inside the body.
Exactly.
Do we know what the triggers are yet? Or is that part of it? We can see that it's happened and now this is how COYA's drugs treat that.
Correct. That's a little bit of a chicken and egg. We don't know exactly what drives all these diseases, but what we do know is that inflammation is a hallmark of all these diseases. And if you address inflammation, you can potentially stop the progression.
So talk to me how you ended up here, right? I know the team's got a tremendous background here. What's your personal story? Why target one of the most difficult areas of medicine to see progress?
Well, the credit for goes to the founders of COYA. I came in 3 years ago into COYA. Howard Berman, our former CEO and current executive chair, founded this company based on a personal passion. He took his dad, who had dementia, to see Dr. Stanley Appel. And that kind of, I'm fast forwarding the story, but that kind of was the evolution of COYA. So it's really their dream, their vision, Dr. Appel and Howard Berman's vision, that I'm kind of carrying the torch forward on. My personal passion was when I saw saw and talked to Howard 3 years ago, I could immediately see the impact this could make on patients' lives.
Yeah.
And not just patients. These diseases have a huge impact on caregivers. So the enormous impact on caregivers and patients, and that's what motivated me to come and say, no, I want to be a part of this story.
And your background prior to being here was in biotech for your whole career?
I pretty much through and through pharma, biotech, kind of boring in that sense.
You're brave, man. No, that's not boring. That's brave. That's one of the toughest industries as investors that we know, but I can only imagine as an operator. So how has your success been prior to coming here, and what gives you the confidence at COYA? Why'd you take this job if you had options elsewhere?
Well, why I took this job is a combination of things. I think my experience perfectly brings to the table what COYA needs at this stage. COYA's platform and technology are super exciting, and I truly believe is going to make a difference for patients. And then the team at COYA is just a fantastic team. My background's all biotech. I like to say I grew up at Bristol Myers Squibb. I spent over a decade there in both scientific roles, running clinical trials early in my career. But then I quickly transitioned more to the commercial side of things. I pretty much did a lot of different roles at BMS, which is a nice thing about being in companies like that. The last 11 years, I really focused on companies where I can go and take my experiences and personally create value.
Yeah.
And that's what I've done.
So you came out from the training grounds of big pharma, understanding the science, what matters to them, what creates value for them long term, and then you went to the other side. You went from, in Wall Street terms, you went from the sell side to the buy side. in a way. So let's talk about the pipeline, the drugs, the most recent developments, because I do believe you had recent news where you just kicked off a new trial. So let's use that to explore what the drugs that are in the clinic right now.
Yeah, so we just kicked off a trial called All-Stars.
Okay.
It's a potentially registrational phase 2 trial in patients with ALS or Lou Gehrig's disease.
Okay.
We've begun dosing. It's a 6-month endpoint. So we really expect to get into the depth of recruitment next year and move that trial forward. The trial's objective is to look and confirm that COYA 302, our lead asset, which is what we're studying in ALS, can make a difference in ALS patients. And if the data is good, which we fully anticipate it being, I think we have a good path with the FDA to get approval. So
you just kicked off that trial. Talk to me about the data that you saw in that trial in phase 1. What are the metrics that you use to evaluate these types of diseases and that you saw the improvements in?
So ALS is measured typically by a score called ALSFRS.
Okay.
What it is it's a 48-point scale score. The lower the score, the worse the patient is doing. It's a quality of life measure, essentially. How well is the patient functioning? So that is our primary endpoint, and that is the FDA-accepted primary endpoint. So in a small trial, in an investigator-initiated trial, this combination of IL-2 and CTLA-4, which is our COYA 302, showed something remarkable. At 6 months, we essentially saw no progress in the disease.
Wow.
These patients were where they were when they came into the trial.
So their ALS scores were exactly where they were. They didn't move. And these are all visibly observable signs, right? So it's not things that it's a number that you can deny because like, oh, it's just a number on a page. You can see it in these patients' actual lives.
You can see it. The patients can feel it. Their caregivers can feel it. And then at 1 year, we saw very minimal decline, only a 1-point decline in the score. To put that in context, so people may say, what does that mean that it didn't decline? A normal ALS patient, based on this huge database called PROACT database, declines about 1 point a month, meaning in 6 points.
These patients should have, after a year, been down 12 points, and they're down only 1.
Yes.
Wow. How many patients were in the phase 1?
Very small. This is 4-patient study.
Okay. And then phase 2, how many patients is it?
120 patients in our phase 2 trial that's ongoing. It's double-blind, randomized.
Any issues with recruitment, or are you finding that it's readily, it's just easy to find patients that are willing to do this?
We have just begun. So far, we haven't had any major challenges in recruiting patients. We don't anticipate challenges in recruiting patients with a disease like this. There's such a high unmet need. There's so few trials going on in this space, but I think we'll have a better sense of how fast we're recruiting sometime next quarter.
So that's COYA 302. What are the next catalysts that investors can be looking for, right? You said this is a 6-month endpoint, so you're just getting the first patients, but it's at least 6 months out before we get data on that. Will you report interim results for that trial? Are there other trials underway in the pipeline that you'll have catalysts for in the next 3 to 6 months?
Yeah, so we will have data from an ongoing investigator-initiated trial with the same combination, the low-dose IL-2 and CTLA-4,
Okay.
in patients with frontotemporal dementia.
Okay.
And we anticipate having a readout of that data imminently.
Okay.
In the next month or so.
And so talk to me, measured on the same scale, an ALS score for that condition for frontotemporal dementia, or is it a different?
No, it's a different scale. It's
Okay.
it's CDAS, FTLD scale. There's a Montreal assessment scale. So they're measured by a slightly different scale, because the difference between FTD and ALS is FTD has a behavioral or a cognitive component to it as well, can affect speech,
Okay.
which ALS is more of a
More
muscular
disease.
Yeah.
All right. So what other catalysts? Talk to me about the rest of the pipeline as well.
Yeah, so COYA 302 is our pipeline within a product. And the reason we call it that is our lead is in ALS. It's already in the registrational trial. We'll start the FTD phase 2 trial next year as well for COYA 302. And then COYA 302 has potential in Alzheimer's disease as well. So we're evaluating and exploring that. And at the right time, we'll kind of move that program
So it's the same
forward.
molecule, but just treating different patients, different indications.
Treating different indications, because the underlying driver of these diseases is neuroinflammation, because these Tregs don't work properly, and we address that.
And was
right?
that
So,
what the Nobel Prize was based upon, identifying that this was the source of these three neurodegenerative diseases, that it was the inflammation for all of them?
a combination of things. So the Nobel Prize was awarded first for the discovery that these Tregs or breaks even exist, which Dr. Sakaguchi did, and then others that have added to that by understanding how these play a role in autoimmune diseases, Dr. Sakaguchi's work, and then Dr. Appel's work in neurodegenerative diseases. And then so a lot of applications for Tregs came out of the work that started in the late 90s.
Yeah.
So,
Let's spend a little bit of time getting into the weeds on the science some, because I think it's helpful to just make it approachable if we can. So Tregs are really the focus here. What's the normal function of Tregs, and then what happens when you see the disease indications that they're not doing?
yeah. So, Tregs are the brakes of the immune system. I'm kind of oversimplifying a little bit, but they're the brakes. So imagine a car. A car can go at 120 miles an hour. If there are no brakes and you're going at 120 miles an hour on a curvy road, that's self-destructive.
Yeah.
And that is what the brakes are there for. So the Tregs make sure that the T cells, the T helper cells, the immune system doesn't go crazy, doesn't go over the speed limits it should, if I may.
Right.
So then it taps on the brakes, which is a regulatory T cell, and says, no, this is a road where you should be going at 30 miles an hour, and do that. And that's called homeostasis, right? Meaning that balancing act between speed and brake.
And this happens naturally as you live your life in a normal immune system where it's functioning properly, responding to different stimulus.
Exactly.
Yeah.
What happens in the disease state, like autoimmune diseases and neurodegenerative diseases, that these Tregs become dysfunctional. The brakes don't work as well, or the brakes stop working.
So they're down-regulated. Is that correct? They stop. They're just correct. They're being suppressed from doing what they're supposed to.
Their ability to suppress the immune function has gone down. Right. So what happens is that the T cells now are unchecked. So this is if I use my car analogy, the car starts running wild and it's becoming self-destructive. In the case here, the T cells then attack their own healthy nerve cells, which then leads to some of these neurodegenerative diseases. Because our nerve cells are spark plugs. We have millions of them. If they misfire...
You're really working this car analogy. I like it. This makes it a lot easier to explain.
Somehow I got on this car thing and I'm just going down that road.
Right. So the nerve cells are the spark plugs that keep everything moving.
And when the spark plugs misfire, engine stalls, engine doesn't work. Same thing happens in the human body. And if the spark plugs completely stops, which is when the nerve cells die, then you start getting real progression of disease. And that's when you start seeing deterioration rapidly.
Okay. That makes sense. Now, let's talk about your molecules, because they're used in combination with IL-2, which has been a well-documented, well-studied chemical. We know how the body responds to IL-2, and you're pairing that with your unique compound. What is that doing to the Treg cells?
And that, Lou, is our secret sauce, actually.
Okay. I tried. I had to ask for it.
You hit on it. So low dose, first of all, the IL-2s that have been used extensively are mostly in the oncology setting at much, much higher doses.
Right, to try and activate T cells to attack the cancer cells.
There you actually want the T cells to go crazy.
You're trying to upregulate and attack. But the problem with IL-2, correct me if I'm wrong, is that it's non-specific, right? It triggers T cells and going after cancer cells and non-cancer cells.
Correct.
Okay.
And so here we're doing the opposite. So IL-2 is a quirky molecule in the sense that at high doses, it does what you just said, which is it actually amps up the immune system. At low doses, it does the opposite. It actually amps up the brakes. So the T regs become upregulated. There's more T regs and their function goes up as well. And the low dose format of IL-2 doesn't exist in the market today. So that's a proprietary to us. We have a proprietary low dose interleukin 2 and we keep it in an aqueous state, which allows us to give this drug subcutaneous. So you can give it like a GLP-1. The patient can self-administer or a caregiver can self-administer.
So you're not on an IV drip for 2 hours like an infusion we see with chemotherapy.
No, so no IV infusions. You don't have to go to a clinical setting. But what happens with this combination, why it is such a unique science, is that when Tregs become functional, but the environment remains inflammatory. So I'm going to go back to my car analogy.
I love it.
You fix the brakes, but if all the fluids surrounding the brake are still crappy, it's a matter of time before the brake stops working again.
Right.
So that's where our secret sauce is. Adding the CTLA4 to the IL-2 keeps the macro environment, the macrophages, the myeloid cells, all of them anti-inflammatory. From a pro-inflammatory state, they go to an anti-inflammatory state. Now, if I use the brake fluid analogy, the brake fluids are also anti-inflammatory. The Tregs will stay functional, or the brakes will continue to work for a sustained period of time, which as long as you have the brakes working, as long as the Tregs are functional, everything else lines up. And that's what is impressive about the data we have seen across 4 investigator-initiated trials, 3 diseases, ALS, FTD, and Alzheimer's. We see that when we restore the function and numbers of Treg and keep them functional, everything else follows. We see that it stops progression of ALS.
So how much is the how many times you have, what's the dosing? What's the regimen? Is it once a week, once a month?
Every 2 weeks or every 1 month. So we're studying both those regimens.
So it is possible that after a certain period of time on the drug that the immune system homeostasis could be restored where they could come off drug? Or is this something that you'd have to stay on it for as long as you have it?
You have to stay on it because we actually have data from that small trial where when dosing was stopped after 48 weeks, those patients do start declining again at the rate that you would expect them to decline, which is 1 point a month. And we also see the biomarkers and the Treg function and the Treg numbers all kind of coming back from not being good, right? It's coming back.
Which then becomes the added benefit of this being subcutaneous because it's a very low friction thing that people can do, right?
Exactly.
Compliance is there, which this is the difficulty in biotech is because there's real patient impact, right? Like that's the goal of it. We're improving lives, saving lives, but then we got to flip at some point to the economic model of it, right? So you start talking to me about diseases that are degenerative, that people can live for 6, 7, 8 years. My grandmother was 17 years, could have been longer. That's a huge market. Because if you're taking a drug over that period of time, those sales will add up. What's the market potential here for, each one of these indications?
Massive, because when you especially start looking at Alzheimer's and Parkinson's, we're looking at 10 billion plus markets here. But let me take the first 2 indications just to give you a context, which are the smaller indications. ALS alone is a 3 to 5 billion global market. Just in the United States, COYA 302 in ALS could easily surpass 1 to 1.5 billion in peak sales.
And you're going to have data sometime in 2026 on that trial, you think?
Well, we haven't guided to exactly when we'll have the data because, as you pointed out, we've just begun recruiting. So I think we'll be in a better position to say when the top line's coming. But I think it's fair to say that with a 6-month readout, you can kind of do the math that it's not going to take 2 years for us to complete the trial.
And I'm doing it only from the investing hat of saying, hey, this is a $3 to $5 billion annual market. This is a phase 2 potentially registrational trial.
So in 2027, we could have a potentially approved product if everything goes right. I mean, so it's not that far away.
And that's ALS. So now let's move to FTD. How big is that market?
A very similar market. So it's almost, you take the ALS market and it's going to be the same. There's no approved drug in FTD. So, that could add more. And same thing in ALS. There are 2 products that are available, FDA approved, but they don't really work sufficiently enough, patients continue to decline. So the opportunity in ALS is pretty big, and in FTD. So if you just combine FTD and ALS, you're looking at a $5 billion, $6 billion market opportunity.
Okay.
You layer on that now Alzheimer's, which will take much longer for us to get to the market with a combination in Alzheimer's than you
get to that 10 billion north.
You're getting to numbers that are pretty.
That's when you're moving on to another company, another opportunity. So we can get into that.
But Alzheimer's also takes much longer to get to the market. And that's part of the attraction of ALS as well, right? Is that you can get to the market efficiently and fast because it's such a high unmet need. There are smaller trials. The endpoints are shorter. All of that means that we can get to that finish line faster.
Well, and you said it's a pipeline in a molecule because if you get there with ALS, you're not changing anything from the actual molecule side. You're just changing the indication that you're trying to treat.
Exactly. And also the confidence in the future indications will just skyrocket once we show good data in ALS, right?
So huge market opportunity. You started to mention it a little bit. What are the competitive, what's standard of care right now in each of these indications?
In ALS, there's really two drugs, riluzole and Radicava, that are used. Neither of them really stop completely the progression of ALS. So despite those drugs, patients continue to decline at 1 point a month. So
really?
when I was talking about the 1 point, it's on top of those drugs, right?
And how much market penetration do they have? Like what percent? I mean, are most patients that get diagnosed with ALS receiving these drugs now?
Most ALS patients will receive one of these drugs for sure. Riluzole is more almost a background, and our trials are on top of these drugs. So
I was just going to ask, so they're doing it in combination with standard of care.
Whatever is the standard of care, the patients can be on it, and the data shows that patients continue to still decline, right? So that's what we're doing, and that
should help from recruitment then, because you're not trying to ask patients to come off the standard of care. You're saying, hey, why not try this? So let's get to the safety of that then too, right? IL-2 in high doses, I know, has some toxicities that manifest. What about low dose IL-2 and your molecule combined with that?
Well, we have seen nothing that concerns us from a safety perspective. The only reaction we saw in our trials is some mild injection site reactions, which is expected when you inject something sub-Q. That's we are using 50 to 60 times lower IL-2 than what you were referring to in oncology.
Yeah.
So intuitively, one does not expect any quirky safety signals. And the other component, CTLA-4, has been studied in rheumatoid arthritis in millions of patients. Millions of patients have taken, and I was part of the launch team at Bristol Myers Squibb for Orencia, a drug that targets is a CTLA-4. And I was the worldwide brand director of Orencia as well. So I understand that molecule quite well.
Yeah.
It's one of the safer rheumatoid arthritis drugs, comparing it to the other agents like TNF inhibitors, right? So we really don't anticipate any surprising safety outcomes in this trial.
So no obvious or real high, risk for the molecule, right? From a safety issue. Correct. Then what are the risks here that you see what keeps you up at night as the CEO thinking you're seeing patients respond positively, but where could things go wrong?
I mean, it's biotech where things can always go wrong there may be some unpredictable things that happen in a clinical trial. But I think what we have done is we have de-risked all of that to a significant extent, because most companies clinical trials get in trouble because of some unforeseen safety issue. We don't see a reason why we're going to see that. Now, can I assure you it won't happen? I don't know, but that's why. But there is no scientific rationale to believe that'll happen. Most companies then fail because their efficacy doesn't work at the end of it. Again, we have seen consistent data where the data is not just delivering enough to show efficacy, it's delivering really good efficacy. Right. That even if we end up halfway, we'll still be efficacious.
Still very good. Yeah, it's better than the standard of care by multiples.
We feel we're de-risked there. And then we're de-risked on the science. I mean, the Tregs are well characterized. The association between dysfunctional Tregs and ALS, for example, is well known. Yeah. That we feel that we are hitting the right scientific targets. So what makes me lose sleep at night is that, execution at this point, right? Are we going to recruit fast enough? Are we going to keep the trial moving on? Or am I going to keep the company well-funded, which fortunately we are? And, those types of things on how do I, as a CEO, enable removing barriers for my team, removing barriers for the patients, removing barriers for the sites, so we can get to that finish line in an efficient manner.
Yeah,
right?
no, that makes sense. I mean, you've de-risked it in a meaningful way across the spectrum here. One thing that comes up, because you have the background of being at Bristol Myers, is it takes a lot. Once you, let's forecast forward. This trial in ALS goes well. You file for registration. You get approved. Now you're a small biotech trying to commercialize a drug. So how do you think about that? Do you think about I know you have a partner in Dr. Reddy's already. No pun intended. How do you see that strategic relationship, or what's the plans as you move forward? Is this go to market alone and commercializing the drug, or do it with a company or get acquired?
Well, with ALS, we have partnered with Dr. Reddy's as our strategic partner. So what's nice about that, Lou, is if you think about a small biotech like COYA, what happens when we have spectacular data is people go, oh, the market actually doesn't always react positively, because they say, well, they're going to have to go and commercialize this product. Now they're going to have to build a sales team. They're going to do all this marketing. We don't have to spend a penny on any of it, because Dr. Reddy's, our strategic partner, is responsible for the commercialization of ALS, the first indication. But what is nice about it is that we have that first indication expense kind of taken off our books already.
Right.
But we've retained the rights to all the other indications.
So they don't have any options or rights to FTD or to Alzheimer's at all?
No, they don't.
Okay. And then let's talk about just the balance sheet as it stands right now. You said you're well capitalized. So there's, where's it at? $40 million roughly in terms of working capital as we speak right now?
Well, what I'll walk you through the numbers rather than giving you a number, because what we reported on September 30th is that we had approximately $28 million in cash.
Okay.
Subsequently, on October 27th, we closed a $23 million registered direct round.
Okay.
So you can kind of do the math.
Yeah.
And then today, we just publicly announced that we received $4.2 million in a non-dilutive milestone payment from our strategic partner,
So by
Dr.
my
Reddy's.
back of the napkin math here, that's about $55 million in capital.
I'll let you,
give or take.
I'll let you do the math rather than me.
So well capitalized, studies well funded. It's now, as investors looking at this, as shareholders, you're focused on the results of the trial that will determine the next value inflection points.
Well, the trial is obviously a significant value inflection point, but there's a lot happening between now and the top line that are value inflection points as well. We'll have this data readout from the investigator-initiated trial in frontotemporal dementia, not too far in the future. We'll have additional preclinical type data coming out of the combinations like GLP-1. We have data coming out from our Alzheimer's study, some of the biomarker proteomics type data that'll also be coming. And the combination really opens the door for business development opportunities as well. Whether it's the GLP-1 or other combinations with COYA 301, the potential for combinations is immense.
Yeah.
The recent CTAD meeting, a lot of people emphasized that neuroscience, neurodegenerative diseases, you have to address them through combinations.
Okay.
So
we didn't really touch on the GLP-1 agonist as a combination potential. Let's talk to that on 301. What does that pipeline drug look like?
Yeah, so that's a combination of our COYA 301 with the GLP-1 agonist, the popular weight loss drugs.
Yeah.
What we have seen preclinically is that when we combine these two, we get synergistic suppression of a lot of the markers or improvement of a lot of the markers, depending on whether, basically good signals that show that together they have a lot of potential in diseases like Alzheimer's.
Okay.
This is preclinical models, both animal model as well as human cell line models. Now, we have not yet moved it to the clinic. We hope to do that ideally in partnership with a pharma company, one of the GLP-1 companies, if not
So have you tested it with all manner of the GLP-1s? Have you gone with a semaglutide, a terzapeptide, and then a retatrutide? I mean, the 1, 2, and 3 targeted receptors, or? No,
we only studied it with semaglutide.
Okay.
And we also had compared it with exenatide, which is one of the old GLP-1s, just as a control.
For the audience, the semaglutide is the most well-known.
It's Wegovy,
Ozempic,
and Wegovy. Yeah. Exactly. It's a Novo Nordisk. And Novo Nordisk had a big readout, the EVOKE trials that came out a couple of weeks ago, where I have always said, and we have always said at COYA, that GLP-1s alone are unlikely to really have a significant impact on dementia. Because that's what we're seeing in our preclinical model. When you have only GLP-1 or only IL-2 for that matter, not necessarily the same impact, but when you combine the two, it's 1 + 1 is better than 2. And we can get to those biomarker suppressive levels that are likely to be clinically meaningful, right? I mean, I can't guarantee it'll be, but based on the biomarker data, the trends definitely point to the fact that, this is likely to have efficacy in diseases like Alzheimer's disease in combination.
So add this all up, and I always ask this, is what's the upside here for the company? You get successful trial data out of phase 2, 302. You've got, ALS that can commercialize. How big could this get?
I mean, how big could COYA get? I think we could be a multi-billion valuation company. And right now, we should probably, based on all the things I've told you, and if you layer to that we have a line of sight through our strategic partnership to non-dilutive cash that has come in and will continue to come in, a clean cap table with no debt, I would say today we should be a 300 to 500 million cap company and not a 120 million company.
Yeah, so at least a 3 to 5x from here based upon just, again, comps, what's in the market, how far you've gone scientifically. Well, listen, I'd love to have you back as we have more catalysts that are reached, and we'll get maybe into more of the science then too, and see if we can extend that car analogy to really help us understand us laymen that don't know much of the science. But I appreciate your time, Arun.
No, thank you, Lou. I really enjoyed being on your show. Thank you.